Right, so this is Mr. Kwok. Okay, this is your first lecture on protein. We'll be starting with amino acids. So looking at the structure of amino acids, all amino acids have the same general structure, right? So there is an alpha carbon, which is the central carbon. The central carbon is bonded to 4 different groups. We have the hydrogen atom, the amino group, the carboxyl group, and a side chain called the R group. Okay, so all amino acids have the same amino group, carboxyl group, and the hydrogen atom bonded to the alpha carbon. Different amino acids only differ in their R group. Okay, so when the amino acids are ionized in water, the amino group and the carboxyl group would be ionized, as you can see here. Okay, so because all the amino acids have the same amino group, hydrogen atoms, alpha carbon, and carboxyl group, right? So all amino acids would contain the same atoms: carbon, nitrogen, oxygen, and hydrogen. And because proteins are made of amino acids, right? So all proteins would contain C, N, O, and H. As for the sulfur, right, it's written in the notes that some proteins contain sulfur. That's because in the R group, right, 2 of the R groups contain sulfur, right? So for the amino acids with these 2 R groups, they contain sulfur. Then proteins that contains such amino acids would also contain sulfur. If the protein doesn't contain these 2 amino acids, then the protein doesn't contain sulfur. There are 20 different amino acids, and because amino acids differ in their R groups, right? So there would be 20 different R groups. Okay, so looking at the R groups in more details, this is the same table that you have in your notes. There are 4 different categories of R groups. We'll be looking at them one by one. First is the neutral, non-polar R groups. Right, neutral, so they are uncharged. You can look at the R group and see that there are no charges, right? No positive or negative signs. And if you look at the R groups, right, almost all of them only contains the carbon and hydrogen, with the exception of methylene, this one, which has a S. So for the neutral non-polar R groups, they are known as, or they are described as being hydrophobic because they are not able to interact with water. Water is polar. Okay, so it takes a polar substance to interact with water by forming hydrogen bonds with water. So for such R groups that are non-polar, they are not able to form hydrogen bonds with water, therefore unable to interact with water. The second group of R groups would be the neutral and polar R groups. Again, uncharged because neutral, right? And if you look at the group here, right, you can see that besides the carbon and hydrogen, right, these R groups also contain either oxygen and nitrogen, with the exception of cysteine, which contains a sulfur. Okay, because of the oxygen and nitrogen being more electronegative, right, this makes the R group polar. Okay, and these R groups are able to interact with water by forming hydrogen bonds. So these R groups are described as hydrophilic. Then we have the acidic and polar R groups. Acidic, so you can see that the R groups are charged, negatively charged. They are negatively charged because they have lost a H+. Okay, so therefore they are described as acidic because they have released a H+ to become negatively charged now. Okay, they are polar, so therefore they are hydrophilic. And the last group would be the basic and polar R groups. So looking at the R groups, you can see that they are positively charged. They are positively charged because the R groups have accepted a H+, right? So they have made the surrounding solution less acidic. So therefore, these R groups are described as being basic. Okay, so again, they are polar, so they are hydrophilic. All right, so just to note, right, you are not expected to memorize the structure of the 20 different amino acids. You are not expected to memorize the 20 out groups. Right? No one will test you a particular amino acid and ask you to draw the out group, for example. Right? What you need to know is, number one, the general structure of amino acid that you can be required to draw. And if we show you the different amino acids or different out groups, then you are expected to be able to know which group of out groups these are and what are the properties of these out groups. All right. Okay, so we move on to properties of the amino acids. Right, so they are colorless and crystalline solids, but they are soluble in water. Right, all of them. So just now we look at some of the amino acids with hydrophobic out groups, right? And we say that they are not able to interact with water, right? It's the out group that is hydrophobic, but not the entire amino acids, right? The entire amino acids still have your N and your O, right? So the overall, right, the entire amino acid is polar. So therefore, they are able to interact with water, they are able to dissolve in water, right? But the out group is hydrophobic for those with the hydrophobic out groups. Okay, so just differentiate, or the perspective is different, yeah? So just now we were focusing on the out groups. Now we are talking about the entire amino acids being soluble. So don't mix up the two, don't be confused. Okay, again, we mentioned already that when the amino acids are dissolved in water, the amino group and the carboxyl group will be ionized. So when they are ionized, right, you see that you have this basic amino group because it has accepted a H+. You have the acidic carboxyl group because it has released a H+. Right, so one molecule of amino acid actually contains an acidic group and also a basic group. Right, so for such molecules with both acidic and basic group in the same molecule, we describe these as being amphoteric. Okay, so the amino acids, right, when they are dissolved in water or in solution, right, they are able to act as a pH buffer. A pH buffer is something that resists the changes in pH or maintains a particular pH. All right. So how does an amino acid do that, right? So when you have an amino acid solution and then you add some acid into this solution, where you're adding H+, right? So what happens is when the H+ concentration increases in the solution, the H+ can bind to the O-, right? And therefore, right, the amino acid is removing the H+ that's added, right, into the solution. So therefore, maintaining the pH, acting as a pH buffer. Likewise, right, when you are adding alkaline into the solution, you are adding OH-, which basically means you are removing H+, right? So when you're removing H+, the amino acid is able to, well, release the H+ that it has gained when it is ionized, so that the concentration of H+ is maintained in the solution, therefore maintaining the pH, right? So acting as a pH buffer. Okay, so when both the amino group and carboxyl group are ionized, right, then this amino acid molecule, also very strangely, has got one part that is positively charged and one part that is negatively charged. Right, so it is an ion, right? It's ionized, but there is no net charge, right? Because there is a positive charge and there's a negative charge. So for such ions, right, they are described as sweeter ions. So amino acids are sweeter ions when they are ionized, and just how we mentioned, right, they are amphoteric because one group is acidic and one group is basic. Right, so these two kind of related to each other, right? And then we look at something called the isoelectric point, which is a pH in which the amino acids would be of no net charge, right? Net charge 0. Okay, so typically, right, for most amino acids, the isoelectric point is pH 7. Right, so if you look at these particular amino acids, for example, right, the R group is neutral, right? So we don't have to look at the out group to look at the charge. When the amino group is ionized and when the carboxyl group is also ionized, then this amino acid is a zwitterion, right? And there's no net charge. So the pH that allows both groups to be ionized would be pH 7. But just how we mentioned, right? When there is lower pH, lower than 7, there's excess H+, that will neutralize the O- here. When the pH is higher than 7, right, then the NH3+ here will release 1H+. Right, so in order for both groups to remain ionized, right, then the isoelectric point, the pH, would be around 7. That's for amino acids in which there is a neutral out group. But for amino acids that have a charged out group, then the isoelectric point will be a bit different. That's when you need to go to your notes, right, on page 5. Then there's a little exercise for you to deduce, right, 2 amino acids that have got a charged out groups. What do you think the isoelectric point would be? Is it going to be above 7 or below 7? So try that before you continue with the SLS lesson. Right.